Nucleus is the most prominent and the most important part of a cell. In animal cells it is found in the center, with exception of muscle fiber cells. But in adult plant cell it is slightly away from the center due to the presence of a large central vacuole. A typical eukaryotic nucleus consists of nuclear envelope, nucleoplasm, nucleoli, and chromatin. Nuclear envelope. Nuclear envelope, also called nuclear membrane, is a double membrane covering which makes the boundary of nucleus. Both membranes of nuclear envelope are separated by a fluid-filled paranuclear space. The membranes are composed of lipid bilayer and proteins. The outer membrane of nuclear envelope is covered with ribosomes and is connected with the membranes of ER. There are numerous pores in nuclear envelope called nuclear pores, which are composed of a specialized transport protein called nucleoporin. These pores regulate nucleocytoplasmic exchange of materials. For example, RNA movement from nucleus to cytoplasm. On the other hand, molecules entering nucleus include proteins, for example DNA polymerase, carbohydrates, for example pentose sugar, required for transcription and replication, signaling molecules, for example receptor steroid complex, which is formed in cytoplasm and enters the nucleus to activate or repress certain genes during steroidal hormones action, and lipids, such as steroid molecules entering nucleus to form receptor steroid complex and influence transcription of certain genes. Nucleoplasm. Nucleoplasm is the transparent semi-fluid ground substance formed of a mixture of proteins, for example histones and non-histones, enzymes, DNA and RNA polymerase, free nucleotide and some metal ions, Mg, for the synthesis of DNA and RNAs. So the nucleoplasm is slightly different from cytoplasm. Nucleolus. It is without any membrane boundary. Well, you may think that if it's without any membrane boundary, then how it stands out as separate area in nucleus. Before knowing the answer, think of an oil droplet which is suspended in water although there is no boundary around it. Similarly, due its specific composition, nucleolus is also present as distinct area in nucleus. Nucleolus may be one or more in number. It appears during interphase and disappears during cell division. Nucleolus is attached to chromatin at nucleolar organizer region or nor secondary constriction, heterochromatin, nor is present on the short arm of chromosome number 13, 14, 15, 21 and 22, acrocentric chromosomes. Nucleolus contains 85% proteins, 10% RNA and 5% DNA. There are two specific regions of nucleolus. One is peripheral granular area, which contains ribosomal subunits, and the other is central fibrillar area, which contains ribosomal RNA and rDNA. Ribosomes are synthesized in nucleolus and transported to cytoplasm via nuclear pores. Chromatin. These are internuclear, long, thread-like thin fibers, embedded in the nucleoplasm. Chromatin net is made up of DNA, histone protein, basic protein, non-histone protein, acidic protein, and RNA. Chromosomes. Greek chromas means color and soma means body. Chromosomes were observed by German embryologist Walther Flemming in 1882 while he was examining rapidly dividing cell salamander larvae. During cell division, chromatin material is converted into darkly stained thread-like structures known as chromosomes. Chromosomes can be best studied at metaphase stage. Structure of chromosome. Chromosomes consist of 1. Two identical chromatids. 2. Centromere, which lies within a thinner segment of the chromosomes, called primary constriction. Two chromatids are held together at a point called centromere. Each centromere has a complex of kinetochores proteins present on the opposite sides of the constrictions. Each kinetochore forms the site of attachment for a single microtubule during cell division. Some chromosomes may also have secondary constriction or nucleolar organizer along the length of chromatids. It gives rise to nucleoli during interphase. There may be a question that why chromosomes contains 2 chromatids and not 1? The reason is that chromosomes are actually formed during cell division and before their formation genetic material has been duplicated, so whenever they are formed they have 2 chromatids. Last but not the least, chromosomes are covered by thin prognathous sheath, called pellicle.